The third line of defense is your specific immunity. This is unique to the individual. It's called adaptive immunity because it depends upon what the individual has seen immunologically as to what this immune system looks like. The immune system is what we call a functional system. All the other systems we have looked at have had organs in them with specific functions. There are no organs that are specific to the immune system. Rather, the immune system is a collection of cells and events designed to protect the body from infection. The immune response is systemic. It's not limited just to the site of the invasion. It's also specific. You respond to a particular invading antigen. This is not nonspecific, but rather very specific in what you will attack. Also, the immune response displays memory. Once the system has seen an antigen and it's recognized it and mounted a defense against it, it remembers that antigen for the future. Should that antigen come into the body again, the immune system will respond more quickly and more strongly. Immunology is the study of the specific immune responses. There are two types of specific immunity. Humoral immunity is a function of the B lymphocytes or the B cells. These cells produce antibodies. Sometimes this is referred to as antibody-mediated immunity. If we think of this as a war, the B lymphocytes are involved in chemical warfare. They produce antibodies that go out, find the enemy, and inactivate it. Cell-mediated immunity is a function of the T lymphocytes or the T cells. The cell-mediated immunity is based on the production of cytotoxic T cells. In our war, this would be the hand-to-hand combat battalion. The cytotoxic T cells go out and directly attach to the offensive cell and destroy it directly. All of your lymphocytes originate in the bone marrow. They become programmed during childhood to be self-tolerant, that is, not to respond to self-antigens, and immunocompetent, respond to non-self antigens. T cells receive their education in the thymus. The B cells receive their education in the bone marrow. Immunocompetent T and B cells will then migrate to the lymph nodes in the spleen. These are the majoring filtering organs for the lymph and the blood. In these locations, they're uniquely positioned to see if anything foreign comes into the body. The cells will be activated when they're exposed to the antigen that they are designed to attack. The humoral immune response involves the activation of the B cells. We start with clonally selecting the B cells. We have B cells that are immunocompetent for different kinds of antigens. When an antigen comes in, it goes around and finds the B cell that can respond to it. This is the clonal selection. When the B cell binds to its antigen, it is now dedicated to producing things that will destroy that antigen. It grows rapidly and it clones, it divides. We get plasma cells, which are going to make antibody, and we will get some memory cells. The memory cells will be stored for later. Now, our first response takes a little bit of time to get going, but we do get antibody and we do get protection. However, if we see that antigen again, we already have memory cells that know exactly what to do. They will almost instantaneously clone into plasma cells, and we'll get a much bigger population of plasma cells, so we get a lot more antibody produced, and we will make some new memory cells. So this second, third, fourth, fifth response will be much stronger and much more protective. Antibodies also are known as immunoglobulins. These are soluble proteins that are secreted by the B cells in response to an antigen. They have two regions on them. They have a variable region and a constant region. The variable region is where the antigen will bind, so each antibody will be specific for its antigen in this region. There are 5 different types of immunoglobulins: IgM, IgA, IgD, IgG, and IgE. This constant region is the same in each class. So all of the IgM are the same in the constant area. All of the IgG would be the same, but the IgG constant area would be different from the IgM constant area. This would be how the 5 major classes would differ in structure. Antibodies protect us through 4 basic mechanisms. First, they can fix complement, and whenever complement is activated, we're going to get cell lysis. Antibodies may bind to their antigen, neutralizing them, making them harmless to us. Some antibodies, when they bind to their antigen, cause the cells to clump up. This makes phagocytosis easier. And some of our antigens are soluble, so when antibodies bind to them, they become insoluble. Now phagocytes can see them and can ingest them. When our antigen comes in contact with our antibody, we can either get neutralization, agglutination, or soluble things may become insoluble and we have precipitation. All three of these enhance phagocytosis. These neutralized substances that are coated with antibody are easier for the phagocytes to find. Here they get a lot more bang for their buck. One bite takes in a lot more dangerous material. And here they can finally ingest something they couldn't see before. The other thing they do is fix complement, and complement will lead to the lysis of the cells, but complement also has some components that initiate inflammation as well as coating up things so the phagocytes find them easier. There are basically two types of humoral immunity. Active immunity occurs when the individual produces their own antibodies. This means memory cells are made and the antibodies can be produced again in the future. Naturally acquired active immunity occurs if the individual actually has the disease. Most active immunity today is artificially acquired through immunizations. With an immunization, the individual is exposed to dead or attenuated organism. An attenuated organism has been weakened so it can't cause illness. Immunizations either give no symptoms of disease or very, very mild symptoms of disease. But the body does make antibodies to this organism and memory cells are made. This means should the individual encounter the actual pathogen, the memory cells will produce antibodies and protect them. In passive immunity, you get already made antibodies from someone else. You didn't make them yourself, so you have no memory cells. Naturally acquired passive immunity occurs when antibodies cross the placenta from mother to child or are transmitted in the breast milk should the mother choose to breastfeed. Artificially acquired passive immunity is when you get some sort of immune serum, like antivenom for a snake bite. Because antibodies are proteins, they do eventually deteriorate, so this is a short-lived protection designed just to get you over some crisis, and it's a temporary kind of protection. You will not have the ability to fend off the same disease again. Looking at our humoral immunity, on the active side, you either get the infection or you get a vaccine. Because you make memory cells, you can make antibodies whenever you need them. On the passive side, you borrow the antibodies from somebody. You have no memory cells. This is a temporary protection, but it is not a permanent kind of protection. The cellular immune response is a function of the T cells. Like B cells, T cells are stimulated to produce a clone when their antigen is recognized. Unlike B cells, they can't bind to free antigen. And in truth, very few B cells can be activated by free antigen. Most B cells have to be stimulated by a helper T cell. The antigen has to be taken in by a macrophage, an antigen-presenting cell. It processes the antigen and presents it to a helper T cell. Now, when the antigen-presenting cell and the helper T cell are bridged like this, they both make a group of cytokines, chemical messengers that stimulate the T helper cell so that it can cause the production of cytotoxic T cells. These are your hand-to-hand combat. They can go out and attack the cell directly. And this is also how most of your B cells are activated. B cells typically go through a T helper cell to be activated to make antibodies. Notice how the T helper cell is centrally located in this process. Without a helper T cell, we would not be able to get cytotoxic T cells, and we would greatly limit our ability to make antibodies with B cells. There are different types of T cells. The helper T cells are sometimes referred to as CD4 cells. These cells are important because they interact with all of the other T cells and the B cells to activate them. They're critical for proper functioning of the specific immune system. The cytotoxic T cells are sometimes referred to as the CD8 cells. These are your hand-to-hand combat soldiers. Once they are activated, they attack foreign cells directly, particularly cells that are infected with viruses, cancer cells, and cells that may have been transplanted to your body in grafts. These cells manufacture and insert toxins in the offending cell and cause the cell to lyse. Suppressor T cells are a particular branch of the helper T cells. They help regulate the responses. We don't need those cytotoxic T cells forever, only as long as we need them to take care of the cell in question. We need to be able to turn them off, and the suppressor T cells are involved in this. And just like with the B cells, we also get memory T cells. These remain after the initial response, and should we see the antigen a second time, we will have a much more rapid, much more protective response. Transplants are simply ways to give people other people's tissue. There are four basic types of grafts. An autograft occurs when the tissue is transplanted from one site to another on the same individual. Since this is your tissue, this is an ideal type of transplant. There's nothing we have to worry about matching. An isograft can occur only if you have an identical twin. Again, this is sort of like having a second set of organs. Your identical twin can donate genetically identical material, so there won't be any different markers on the cells. An allograft is when we have tissue transplanted from the same species. This is the most common type of transplant. We transplant kidneys, lungs, hearts, things like that. This is going to require tissue matching because remember, each individual is unique in the markers that are found on their cells. And xenografts are when we transplant from different species. We have varied results depending upon what we try to transplant. We can very successfully transplant pig heart valves or cow heart valves without any problem, but entire organs always fail. Because transplanted tissue will have different markers on it than your tissue, we always have to worry about rejection. The first thing we do is make sure the blood groups match. A-positive people can donate to A-positive people. Then we have to check all of the cell markers. These cell membrane antigens have to match at least 75%. Once the transplant has been done, the patient will receive various kinds of immunosuppressive therapy. Corticosteroids will suppress inflammation. Cytotoxic drugs will kill actively dividing cells. That might be immune cells that are trying to mount an attack against the new tissue. Radiation therapy is usually done and immunosuppressive drugs are given. All of these things tend to weaken the patient's immune system, as it should because we're trying to tolerate somebody else's tissue, but it does make the recipient more prone to infections. Basically, there are three categories of immune system disorders. We have hypersensitivities. These are your allergies. Here the immune system responds inappropriately to something that's not threatening. Immunodeficiencies are when the immune system fails to respond fully to a threat. And autoimmune diseases occur when the immune system responds to self-antigens inappropriately. Hypersensitivities, or allergies, are abnormal immune responses to non-threatening molecules. We tend to call these antigens allergens. A lot of these are haptens. They may react with a normal body molecule, and that makes them allergenic. Typically, hypersensitivities are localized at the point of contact with the allergen. Immediate hypersensitivities are an antibody-mediated response. The allergen is usually something fairly common in the environment, dust, pollen, latex, things like that, that cause the release of histamine. You will get a localized inflammatory response. The area where the allergen contacted the body will suddenly become inflamed. This means we'll have swelling of the tissue as well as heat and redness, but usually it's the swelling that causes the problem. Immediate hypersensitivities can be treated with antihistamines to try to block the action of histamine, and then people can try to avoid the allergen. In anaphylactic shock, we have an immediate hypersensitivity, but the allergen gets into the bloodstream. This would be something like bee venom. Because there is such a massive release of histamine throughout the body, this will cause a massive release of histamine throughout the body. The smooth muscle in the trachea is particularly sensitive to this histamine, and so it will begin to swell. As it swells, it will block the airway. If these people are not treated fairly quickly, they may suffocate because their airway will close off. This is treated with epinephrine to reverse the effects of the histamine. Most people who know they have this type of allergy will carry an EpiPen. The EpiPen is just a stopgap. It's to get them to the emergency room so that they can be further evaluated. People who are subject to anaphylactic shock should really try to avoid their allergen. With delayed hypersensitivity, the allergen diffuses through the skin. These are things like poison ivy, cosmetics, soaps, latex. Usually these act as haptens, attaching to some normal protein in the body. Instead of histamine being released, we release lymphokines. Lymphokines will activate the T cells. So while immediate hypersensitivities are antibody-mediated, delayed hypersensitivity are T-cell mediated. It takes longer for the T cells to be activated, so there's a longer response time. That's why you don't see the effects of the allergy for several hours. This is treated with corticosteroids to suppress the immune system and, of course, avoidance of the allergen. In immunodeficiencies, the body is unable to respond to the demands of the immune system. Severe combined immunodeficiency disease, or SCID, is a very rare congenital disorder. Individuals with this disorder do not make either B cells or T cells. This has been successfully treated with bone marrow transplants and umbilical cord stem cell transplants. If you've ever heard of the boy in the bubble, this is the condition he had. These people can mount no immune response whatsoever, and if they're not treated or isolated, they very quickly die of infections. Acquired immune deficiency syndrome, or AIDS, is an infection with the human immunodeficiency virus, or HIV. This particular virus infects the macrophages and the helper T cells. You will remember that the helper T cells are critical to activating the entire immune system, so this cripples the immune response completely. Patients with this typically die from opportunistic infections. An opportunistic infection is an infection with something that shouldn't make you sick. Things that we experience all the time, but if you have a normal immune system, you totally ignore it. There is a difference between being HIV positive and having AIDS. If you're HIV positive, you have the virus and are capable of giving it to others. You have AIDS when your CD4 count goes below 200 and you start having opportunistic infections. With autoimmune diseases, the immune system attacks self-antigens and damage normal tissue. We don't really know what causes autoimmune diseases, but there are some theories. One is that the lymphocytes are inefficiently programmed at the beginning. They don't learn self from non-self completely. Another idea is that there are gene mutations, that over the course of our life we may develop some new proteins that show up on our cells, and because our lymphocytes learned about our proteins early on, they don't recognize these as being self-proteins. And then finally, there's the theory that there is a cross-reaction between antigens, antibodies, and self-antigens, that somehow we get some naturally occurring antigen or some antibodies that respond to a self-antigen, and this causes a bigger complex that causes the immune system to activate. There are several diseases that are classified as autoimmune disorders. In multiple sclerosis, the immune system attacks and destroys the myelin sheaths around the nerves. In myasthenia gravis, the neuromuscular junctions between the nerves and the muscles are damaged. This occurs particularly in the muscles of the face and neck. In Graves' disease, the antibody that's produced attacks the thyroid gland, causing it to overproduce thyroid hormone. This will ramp up the metabolism to dangerously high levels, and heart failure can occur. In type 1 diabetes mellitus, the cells of the pancreas that produce insulin are destroyed. Systemic lupus erythematosus is a very complex autoimmune disease. Typically, the antibody is against DNA. However, the DNA can be in different kinds of cells. If the skin cells are the target, then the individual will have unusual rashes and skin problems. However, if the DNA of a major organ, such as the kidneys or heart, is targeted, these organs may fail rapidly. Rheumatoid arthritis is when the joint tissues are the target of the autoimmunity. Here the tissues are destroyed, scar tissue forms, and the joints become deformed.